Hello everyone, my name is Agues-Barbosa. I'm a master's student of psychobiology here in Brazil. And today I'm going to present to you the preliminary data from my dissertation. And first of all, I would like to thank the organizing committee for selecting my work. It's a great pleasure to present for you guys. And I would like to dedicate this presentation to the frontline professionals, which are right now putting themselves out there, risking their lives and their families' lives for us to be home. So thank you, bus drivers, thank you, doctors, nurses, thank you, supermarket workers, and everyone which is working hard for we can pass through this pandemic as well as possible. So thank you very much. And my research project is Untitled Behavioral Differences on Acute Alcohol Exposure in 3 zebrafish strains. And it was developed by me and by my advisor, Ana Carolina Lucchiari. So let's begin. So alcohol is one of the most consumed psychoactive drugs in the whole world. It just stays behind of caffeine, obviously. And this is partly because alcohol has a low cost and is easily to access. Besides that, alcohol is a legal and socially used and encouraged drug. So every time that we have a meeting with our friends, it's almost certain that alcohol will be involved with those celebrations. And this is a problem because almost 43% of world population consumes alcohol regularly. And in worldwide, almost 3.3 million of deaths are related to alcohol. So alcohol kills more than AIDS and tuberculosis, for example. Alcohol is this small molecule, which is soluble in water, so our body doesn't have so much trouble in metabolize it. Alcohol acts in a lot of systems in our organism, since liver, kidneys, and our central nervous system. And in our brain, alcohol presents this biphasic effect in which we have an initial state of excitation followed by a state of depression of the system. And this is due because alcohol interacts with a lot of neurotransmitters, including GABA, glutamate, dopamine, and serotonin, and a lot of other neurotransmitter systems. However, as we know, not everyone becomes dependent of the drug. This is partly because there are individual differences influencing the way that people or individuals are going to respond to this drug. And this is clear when we have a group of friends and one of them, we always have a friend that is more sensible to the alcohol. They need less drinks to change the behavior, while others are more resistant to this drug. And it would be great if we could test this in humans. However, we know that there are a lot of ethical limitations in the study with our species, so we need to find other ways. And zebrafish is an excellent model because there are already in the literature that we have individual differences in these species, and they are related to how individuals are going to respond to alcohol. Those 2 papers were published by 2 students by Fish Lab. And those individual differences, they happen due to a lot of factors, a lot of characteristics. One of them is genetic background. So how much the differences in genomics contributes to those differences that we see in the population. And one of the manners that we have to test this is to compare strains from a same species. So zebrafish, for example, we know that there are a lot of strains that are vastly used in a lot of laboratories around the world. Here we have this paper which shows that we have morphological differences between the strains. So for example, here, the 2 strain, they have these bigger eyes, they have the bigger head and the different craniofacial formation when we compare to the other strains. And when I decided to work with this in my master, I started to look into the literature to find if there were already papers that show the differences in the strains related to alcohol response. And actually there is a few works talking about it, investigating this. The problem is those works, they do not have a robust response. So we don't know actually yet which strain is more sensible or more resistant. So we need new approaches to understand this better. So having all of this in mind, the aim of my research was to investigate how a single exposure to alcohol affects locomotor and anxiety-like behavior of 3 zebrafish strains. And how did I do this? I did a simple novel tank test and exposed animals to acute alcohol concentrations. So here is my experimental delineate. I had 15 animals from each strain. So although all of those 3 strains are considered as wild type, I prefer to call it just AB, wild type, and TU, just to make it simple. And I exposed 15 animals from each strain to one of these three alcohol concentrations. So I had alcohol control, 0%, which fishes were just exposed to water system. I had ethanol 0.5 and ethanol 1%, and fishes were individually exposed to these concentrations for 60 minutes. And during this time, I recorded behavior and analyzed video in animation. So here are my results. Just to be clear, I did a comparison between strains and intra-strain. So for example, low cases here, they indicate differences inter-strain. So that's when I'm comparing, for example, white type fishes that were exposed to 0% with TU fishes that was exposed to 0%. And this little symbol, this little star here, it indicates the differences intra-strain. So that's when I'm comparing, for example, white type fishes that were exposed to 0, 0.5, and 1% of alcohol. I hope that's clear for you guys. So here I have the 2 first behavior measures, the distance traveled and mean speed. As we can see here, in both behavior measures, the TU fishes differed from AB and white type fishes when they were exposed to 0% of alcohol, which means that when we have a baseline behavior, they already present changes between the strains. And other interesting thing is that in both parameters, TU fishes only showed behavioral changes when they were exposed to 1% of alcohol. Other thing is that for both behavior measures, AB fishes presented more variation intra-strain when you compared AB fishes that were exposed to 0, 0.5, and 1% of alcohol. They were more variable than the other 2 strains. And for maximum speed, we see the same pattern. Both AB and white type fishes, they were more variable than TU fishes. And here is when the thing becomes interesting, because we have the 2 anxiety-like behavioral measures, the distance from borderline and time immobile. Here we can see that as locomotor behavior, TU fishes, they differed from white type and AB fishes already in control group. So they were a little bit more anxious here, as you can see, when we compare the 2 white type and AB fishes. Other interesting thing that here is really interesting, because we expect that the higher the doses, the higher the anxiety of the fish. However, we don't see this for TU fishes. They showed the completely opposite behavior when we compared to white type and AB fishes. Here we can see that the higher the doses, the more relaxed the fishes were. And for white type fishes and AB fishes, that was the opposite. The higher the doses, the more anxiety-like behavior they showed. And here we can see that as locomotor parameters, TU fishes only showed behavior changes when they were exposed to 1% of alcohol. I'm going to try to discuss this in the future. And as we saw for the other behavior measures, both white type and AB fishes were more variable when we compared the behavior intra-strain, when compared the strain that was exposed to different alcohol concentrations. And okay, so what can we conclude of all of this? First of all, that as we saw in other papers, our results confirm these previous findings that zebrafish actually have present behavior differences when they are exposed to alcohol. And between the three strains, the TU fishes seems to be more resistant to alcohol. We don't know why that happens. I need to perform new experiments to understand this better and look into the literature as well. And AB and the farm-raised white type, they presented high variation inter-strain when we compared to TU fishes. And these papers, these results, they can be important when we try to develop alcoholism treatments, because we need to take into account the differences that there are in the population. And for my next steps, which I'm hoping that I can accomplish this year, we are going to perform a molecular analysis. So I'm going to look into the genetic variation per se. And I'm thinking about adding intermediate alcohol concentrations like 0.1% and 0.25%. And lastly, which is another aim for my dissertation, is to test the effects of alcohol through ontogeny in between these strains. So, well, that's all. I hope you enjoyed my presentation. If you have any concerns or questions, you can ask me, or you can send me an email. Thank you for the organizing committee. Thank you for Fish Lab for the supporting, and stay home if you can. Thank you.